Yes, well, welcome everybody to the Darwin Festival, Shrewsbury Festival 2021. My name's Torrin Morgan. I'm the head of biology at Shrewsbury School, Charles Darwin's alma mater, of course, and we're delighted to be bringing you this lecture. Before I introduce tonight's speakers, please can I just note that this session will be recorded and that there will be a question and answer session at the end, and any questions that you might have should be submitted via the question and answer function. In The Origin of Species, Charles Darwin established a scientific basis for understanding how evolution occurs by natural selection. Since Darwin's time, understanding insights from the field of genetics, behaviour and ecology have continued to illuminate how and why species evolve. In this talk, Professors Grant and Grant will discuss the progress that has been made in their understanding of speciation, with special reference to the young radiation of Darwin's finches on the Galápagos Islands. They draw upon the results of a long-term field study of finch populations spanning several decades, combined with laboratory investigations of the molecular genetic basis of beak development and variation. Professors Peter and Rosemary Grant of Princeton University have been studying Darwin's finches on the Galápagos Islands since 1973, and their research has been published in 4 books, most recently, How and Why Species Multiply. Professor Rosemary and Peter Grant, it's a great pleasure to welcome you, albeit virtually, to Shrewsbury. We wish you could be here in person with us tonight, but if I can now hand over to you, and we look forward to hearing your talk.
Thank you very much for that introduction, and also for inviting us to participate in what looks like a fantastic week of celebration. Oops. So, happy birthday, Charles Darwin. And thank you for joining us in this virtual world of ours. Cheers. Now, Charles, you made fundamental contributions, as we've just heard, to our understanding of the biological diversity on this planet, and asking several fundamental questions and giving us guidance on how we, your successors, should proceed to go further in our understanding. And I don't know whether you remember this quotation that comes from you. You gave this guidance: those forms which possess in some considerable degree the character of species, but which are so closely similar to some other forms, or are so closely linked to them by intermediate gradations, that naturalists do not like to rank them as distinct species, are in several respects the most important to us. Well, there are groups of organisms like that that are particularly suitable for study. We call them adaptive radiations. Excuse me one moment. Here we go. We call them adaptive radiations because from a common ancestor, one or a small number, a multitude of species have been formed, adapted to different environments. These are the cichlid fish, perhaps the most spectacular example of adaptive radiation, that live in the Great Lakes of Africa, hundreds of species within one lake. There are other striking examples too. The anolis lizards, for example, in the Caribbean, about 150 species of them. And then in the Hawaiian archipelago, there are honeycreeper finches, about 50 species, varying in color and beak shape and size, pattern and form. Now, we have followed in your footsteps, Charles, by going to the Galapagos and studying a group of little birds called finches, and named after you, Darwin's finches now, because you were the first to describe them and make observations of their feeding behavior. It's a young radiation, approximately 1 to 2 million years old only, compared with much older ones. They live in a pristine environment, at least some islands are in pristine condition, and their environment is very dynamic. They're famous not only for the fact that several species have been produced, but famous for some unique behaviors. Here, for example, is one species known as the woodpecker finch using a tool. Very unusual, not unique quite, but very unusual behavior. Then here is another one, different species, that has developed the habit of pecking at the back of seabirds, drawing blood and drinking it. And those same birds on the same island also exploit the eggs of those seabirds by pecking at them, breaking them, and eating the contents. And if breaking them that way doesn't work, they kick them against a rock and smash them open. So the islands are very dynamic in origin and in location. They are volcanic out in the Pacific, and they exist on a plate, a geophysical plate that is proceeding very slowly towards the South American mainland at the rate of approximately 60 kilometers per million years. And in that period of a million years, the Galápagos have experienced 10 cycles from extreme glacial conditions to extreme interglacial conditions, with effects globally on sea levels. And the islands, therefore, have experienced a fluctuating sea level that has caused them to increase in size, increase in height, and become more close to each other with the dropping of the sea level. So it goes like that, and the ultimate islands have coalesced. Excuse me. I'm going to go back to show you what it's like with the interglacials being like it is at the moment, with islands well separated in space. So we think geophysically and climatologically, these islands are exceedingly dynamic, but they're also dynamic in contemporary time as well. How did the species arise in such a short time? Well, you, Charles, had put your finger on it by expressing the key idea about how two species evolved from one. It's a process of lineage splitting. The split is facilitated by spatial separation of the two daughter populations, and they reach a point eventually in time where they're no longer capable of interbreeding, and so the product is two species produced from one. Now I'm going to put your argument into modern language with a little diagram to explain the steps. The first step is the arrival in the island by dispersal from the mainland of a colonist species. Populations on other islands are established through dispersal, and in those other islands, the populations adapt to the local conditions because those conditions are not the same across the archipelago. And then the third phase in what we call a speciation cycle is the coming together of previously separated populations, and now natural selection acts in such a way as to minimize two things. It causes divergence that minimizes competition for food and also minimizes the likelihood of interbreeding, assuming that the products of interbreeding are relatively unfit. That is, they don't survive very well or they don't breed very well. They're infertile. Now that process of divergence, where separated populations come together and are driven apart by natural selection, is sometimes referred to as character displacement. And I'm going to give an example of that with our study on the little island of Daphne, right in the middle of the archipelago. That is the island. It's small, it's 3/4 of a kilometer long, 120 meters high, and it's never been settled by humans. The current occupants are 4 species in the genus Geospicer, or ground finches. There's a small, a medium, and a large species. And at the bottom right, a cactus finch with relatively elongated beak. I'm going to interrupt the scientific narrative for a moment and let you visualize what it's like to be on the island. First of all our supplies are put into quarantine, water, food, clothes, prior to arrival on the island. Then we disembark. In the middle of the island is a crater. The birds fly everywhere, and we have to go everywhere to follow them. We sleep in a tent. We cook in a cave. We put up nets to capture the birds. They're unharmed. We take them out. We withdraw to a cave, a shady cave, and there we measure the birds. We put 3 colored bands and one ring, one numbered band or ring on the leg, so that we have a unique combination. And when we release the birds, we can identify them as individuals and know what they're feeding on and what their sizes are. Before we release them, Rosemary takes a small drop of blood for later DNA analysis. This is rather like nurses do with newborn babies, taking a blood source, little spot of blood for DNA analysis and blood typing. So the 4 species in the archipelago have many populations, and each one of these dots refers to the average beak width on the vertical axis and beak length on the horizontal axis of the 4 species, the small, the medium, and the large ground finch, and this is the cactus finch. The principal species are Fortis, the blue ones here, and this is the Daphne population, and Scandens in red over here, with population sizes, breeding population sizes, on average being about 150 pairs respectively. They differ in feeding ecology. Fortis, the medium ground finch, is a generalist. Now, having banded a lot of these birds, measured them, and watched what they feed on, we know that birds that are seen feeding only on small seeds have small beaks, and those feeding on large seeds have large beaks. So the species is a generalist, but there's some degree of specialization on food type. Scandens, on the other hand, is a specialist. All birds do the same thing. They all probe into Opuntia cactus flowers for the pollen and nectar rewards. They all, with their long beaks, hammer a hole in a fruit, top left, and extract seeds, which they then crack and open to get at the kernels within. Now, are these species evolving now? And if so, how and why are they evolving? And what does that tell us, if they are evolving, about speciation? Well, they certainly are evolving. Viewed across the 40 years of our study, we have observed 2 periods of intense natural selection against a general background of no change from year to year. Those occurred in 1977 and 2004. The circumstances were different, and I'm going to discuss each of those in turn, but there is a common denominator, and that is a drought. A drought that converts vegetation like this into that. So profound food shortage and lots of individuals dying as a result of starvation. The first of those episodes occurred in 1977 when the rains practically failed altogether. And the average beak size of the Fortis population increased throughout 1977 at a time when 80% of the population died. Now, the reason for that is that after the supply of small and soft seeds was depleted in the early part of the year, finches had to turn increasingly to the large and hard seeds remaining in the environment, including this one up in the top left. The icon is a photograph of tribulus, a woody fruit, which has to be cracked in order to expose the small seeds inside. Only the large beak birds could crack those open. They survived, and the small ones died. And so natural selection had occurred. Now, for evolution to occur, there has to be a change in the genetic characteristics of the population from one generation to the next, which means that the trait subject to natural selection must be genetically heritable. And it is. And we know that from a very strong correspondence between offspring sizes and parental sizes of beak size of the 2 species. And that resemblance arises through the transmission of genetic factors from parent to offspring each generation. So evolution did occur because when the birds the survivors started to breed in 1978, following the resumption of rains and the regeneration of the food supply, they produced offspring, shown here, very similar to themselves as a result of that inheritance of genetic factors. So evolution occurred as a result of natural selection on a heritably varying trait for reasons that we could understand when the environment changed. The next event occurred many years later, but the key preceding event that potentiated it was an enormously wet year associated with the El Nino phenomenon that occurs in the Pacific. 1.3 meters of rain fell on this semi-desert island in this extraordinary year. And according to Coral Core data, the El Nino event was the most severe such event in 400 years. So we were extraordinarily lucky to be there to witness it. And what does it look like? What did the effects look like? Well, here's a view of the island in a normal dry season. And when it rains, the rain is seasonal, the island turns green. And in the foreground are the plants tribulus that I mentioned earlier, producing the large and hard fruits that made the difference between survival and non-survival in 1977. Well, the abundant rain that fell over a period of 8 months generated an enormous amount of plant activity. And so the feature was completely transformed into something like a herbaceous jungle. Foremost among the plants growing were vines. They grew everywhere. They grew over the cactus bushes. They grew over trees. They grew over each other. And even in the following dry season, the next year, we could see the remnants of their effects, draped vines on the bushes and the trees. Now, in 1983, when this was going on, 2 female and 3 males of a different species, the large ground finch, that had immigrated to the island and in other years would have probably gone back to their own island of origin to breed, stayed to breed instead on Daphne and established a new breeding population. And so their numbers built up very slowly. So by the time the next really severe drought occurred in 2004, there were more than 150 of these birds on the island. And you can appreciate by looking at this lower picture what massive beak and powerful musculature this bird must have, making it a very efficient cracker or cruncher of these tribulus fruits. And so they were at an advantage as the number of birds declined and the food supply declined. These ones in the top right, Magna rostris, had an advantage over the larger members of the Fortis population. Here's the smaller members of the same population. These ones outcompeted those for these fruits, and these ones therefore suffered a big decline. They were not eliminated altogether. They were dying at a faster rate than these that were also dying, but as put it the other way around, these were dying at a slower rate than these ones. So the difference between this population and that population became magnified as a result of a decline, a severe decline in the average beak size of the Fortis population. And it stayed there because of the inherited nature of beak's size. That level stayed there and didn't return for the rest of our study. So this is an example of character displacement, as I had mentioned at the beginning, the coming together of 2 populations and then diverging as a result of natural selection, in this case, minimizing competition for food on this little island of Daphne. So we have a very good understanding of the relationship between the environment and beak size. What about the genetic factors underlying beak size variation? Well, to address that major question, we collaborated with molecular geneticists in Sweden. Leif Andersen has a very large group in University of Uppsala. His lab ran an investigation into finch DNA. Sangeet Lamachani was the man on bottom left here who spearheaded the attack. And they found two genes, ALX1 and HMG2, that affect beak development. ALX1 is a transcription factor affecting beak shape, and HMG2 is another factor affecting beak size. So this is the variation we were trying to understand, and having discovered those two genes. And now I'll explain briefly how they found the genes. They first sequenced every species and many individuals of each of those species to arrive at a genomic profile of each one. And then they clustered the species into size groups of small species, medium species, and large species, and aligned the genomic sequences in pairs of contrasting size so that the points of a major difference between the contrasting groups could be identified. And this is what it looks like in diagrammatic form, with all the colors referring to genetic positions. You see a pair of contrasts between, in this case, large and small, large and medium and small ground finches and tree finches in these contrasting arrangements by size. So running along the genome, one can, by this means, identify those locations where the contrasting members of a pair differ most. And it turns out that there's one position for each of the contrasting pairs. It has 4 genes, one of which, HMG2, is known to affect size variation in us humans, as well as dogs and cats and rats and rabbits. Now, in Darwin's finches, they affect beak size. So variation at this locus is associated with variation in beak size. What we call the LL genotype is associated with large beaks, and the SS genotype with small beaks, and intermediates associated with intermediate beak size. And the interesting thing is that in that character displacement example that I have illustrated a few moments ago, selection acted not only on the beak size itself, but also to the underlying genotype. So, for example, the SS genotype survived particularly well. Those individuals with the LL genotype survived really poorly, and intermediate survived to an intermediate extent. Very, very strong selection at the genetic level, as well as the phenotypic and measured level. So I'm going to summarize at this point and draw 2 major conclusions and then a third one. First of all, natural selection occurs when the environment changes. Secondly, other species determine the direction of evolution, up or down, in our case. And both of these are core ingredients of the classical Darwinian mode of speciation. The first conclusion applying to the first phase of divergence, and the second one applying to the interaction between species in the second phase. Now I'm going to hand over to Rosemary. This is the easiest point to get onto the island.
Okay, so we started off with 2 species on the island, and now we have 3, all ground finches. And what I'm going to do in my part is to just introduce at the beginning, how do these 3 closely related species stay apart? There is occasional hybridization and gene flow between a couple of them. And I'm going to ask what are the evolutionary consequences of rare gene flow between the species? And then finally, I'm going to end up with one of the most exciting parts of our study, and that is the origin of a completely new lineage, which we have followed from its inception right through 6 generations. So first of all, how do the species keep apart? Well, they're all ground finches, and all Darwin's ground finches are similar in plumage, so we can't use plumage. Males are black on the left here, and females are brown. They all build similar nests, and they all, as far as we can tell, have similar courtship behavior. But they differ in song, and they differ, as Peter has shown you, in size and in beak shape. And it turns out that, well, first of all, when we went to the island, we said we can discriminate the individuals in this way with song and appearance, morphology, but we had to ask, can individuals of the birds discriminate between their own and another species? First of all, by appearance, or as we call the same morphology, and secondly, by song. And the way we did this is we took a female stuffed museum specimen of a tortoise, put it on one end of a pole, and on the other end, we put a stuffed female specimen of scandens, took it into the territory of a living territory owner, and asked, could the territory owner distinguish its own female from another species female? And the answer was a resounding yes. It was during the breeding season, and they vigorously courted a female of their own species, ignored the other one, even though it was just a stuffed museum specimen. And we did this many times, and we did it with controls. And then what we did to find out whether they could discriminate just using song without any morphological cues, we played back Fortis song, first of all, and Fortis came into the loudspeaker, Scandens completely ignored it, played back Scandens song, Scandens came into the loudspeaker, and Fortis completely ignored it. So they could discriminate on the basis of appearance and song in exactly the same way that we could do so. And it turns out that song is really important. The song is very different between the species. This is a Fortis song, it's a modulated song. This is a Scandens song, a series of repeated notes. There's individual variation within the species, but it's always on a species-specific song theme. There's another thing we know about song, and that is that it is, again, this was done, first of all, in captive birds by Berman. We found that song is learnt from the father during a very short receptive period early in life, between day 10 and day 40. This corresponds to the last few days when the birds are in the nest and when the birds are out of the nest being fed by the parents, and all this time the father is singing. So it's not too surprising that males learn perfect renderings of their father's song. Females don't sing, but we've been able to show by the pattern of breeding that they also learn their father's song. So once learned, this song is retained for life. Life can be as long as 17 years. We record every bird every year, and we know that they don't change their song. And as adults, they pair according to the species song that they learnt early in life. So this acts as quite a robust pre-mating barrier. But after all, it is based on learning. So it is also vulnerable to disruption if a young bird hears and learns the song of another species during its sensitive period. And this happens very occasionally, but it does happen. And one typical way it happens is when the father dies, the mother that doesn't sing is left to raise the offspring. And if in the neighboring territory there is a bird of another species singing away, then the young will learn that species song. So in this way, it learns the wrong song. Now, the colonization of Magnorostris that Peter told you about gave us an opportunity to test this. Magnorostris is a bird of almost twice the size of Fortis. It's very aggressive, a really robust bird with a very loud song. And over the years, we had 8 Fortis and 2 Scandens that learnt and sang a Magnorostris song. So here's a Magnorostris song down here. Here's one of the Fortis that sang a Magnorostris song. A normal Fortis song is here. one of the Scandins that sang a Magnarostrus song, normal Scandin song, is here. So they learned this song when they were young. And then when the breeding season came around and these little birds opened their mouths and sang a Magnarostrus song, a Magnarostrus whipped in as if from nowhere and just beat the little bird up. And so it never survived to breed. So it seems that when the size difference is large, this barrier is robust. Even learning another species' song doesn't lead to hybridization. However, it's a very different story when the size difference between species is small. So there's Scandins at 22 grams, Fortis approximately 17 grams, not very different. But in this case, learning another species song can lead to hybridization. Once again, we have very, very few birds, maybe 1% of the breeding population learns the wrong species song. But we had about these few birds which we followed in the first 10 years of our study. And we wanted to see if there was any evidence of genetic incompatibility, because Fortis and Scandins are not each other's closest neighbors, even though they are ground finches. So we wanted to know how fit these hybrids were they viable? And for the first 10 years of the study, we had none of these birds survived the dry season. The dry season at this time was extremely dry. So it could have been that there was no genetic incompatibility and it was just food that was in short supply, which is why they didn't survive. And this seems to be the reason, because after 1983, when the island, in this incredible El Nino event, as Peter said, the strongest in 400 years, the island was completely transformed from a large hard seed producer to a small soft seed producer. And under this abundance of food, these hybrids began to survive. But we could still ask, how well did they survive compared with pure species? So the first time we looked at this was in 1983, all the birds born in 1983. And at this time, we had a large number of birds. We had over 1,000 Fortis were born that year, and over 550 Scandins were born that year. And we had 12 hybrids that we could follow. And we found then that when we looked at their lifetime survival, that the hybrids actually were doing rather well. They survived as well as, if not better, than the pure species. And the same happened repeatedly. So once they survived, how did they mate? Well, they followed the rule. They mated according to the song that they had learned early in life. So if there was a Fortis Scandins hybrid that had learned a Scandin song, it would mate with the Scandins. So we had genes flowing, just a small trickle of genes flowing both ways, from Fortis into Scandins and back from Scandins into Fortis. But for the next 30 years, any trickle of gene flow that was going nearly always was going from Fortis females into Scandins males. And the reason for this is that the sex ratio in Scandins at this time was very much skewed in favor of males. So there weren't really very many Scandins females to go round. And any Fortis Scandins hybrid that had learnt a Scandin song mated with Scandins at this point. The other thing is hybrid males were produced, but the hybrid males were smaller than the Scandins males, and they were outcompeted both for territories and for females. So because of this, this is where hybridization started here. And when we looked at the change in beak shape across these 40 years, we found that in the last 30 years, the average beak shape was getting blunter and blunter and blunter and much more Fortis-like. So we then wanted to know, we had actually tracked the genes going across, but we hadn't tracked specific genes, and we wanted to know whether there's a specific allele that could have contributed to this change in beak shape. And as Peter told you, we sent the blood samples over to Sweden, to Leif Andersson's lab, and they found this wonderful gene called ALX1. Now, this is a gene that is a transcription factor. It's also one that is known from humans, because in humans, a mutation in the ALX1 causes cleft palate in humans. So we knew that it was involved in cranial facial development, and we found that the ALX1 blunt allele was associated with Fortis blunt beak, the ALX1 pointed allele associated with Scandin's pointed beaks. And we could follow or track through our blood samples, the ALX1 blunt allele going across into Scandins, which resulted in the Scandins bill getting blunter and blunter and blunter. But of course, there's a large amount of variation that we saw. And so all these are Scandins. They were all taken out of the same net in 2012. The one at the top has a very strong pointed bill and has measurements that are exactly comparable with measurements of Scandins before there was any hybridization. These two bottom Scandins have much blunter beaks, and they have Fortis genes in them and have much blunter and more Fortis-like. So the population now is very variable in bill shape. And we have seen how the genetic and morphological variation had increased. So we wrote in a paper that a mosaic genome arising from hybridization between two species generates genetic and phenotypic variation on which selection can act. And we suggested that in a new or changed environment, this could lead to a rapid change, perhaps along a different trajectory. And we never actually thought we would see it happening, but we did. And we saw it happening in this formation of a completely new lineage of finches. This happened at a time when we had a lot of birds on the island. There must have been 800 to 1,000 birds on the island, all of which were banded. And then this bird suddenly appeared. It looked like a blown up Fortis superficially. When we caught it and measured and weighed it, it was 28.5 grams. A normal Fortis is 17 grams, so considerably larger. We banded it 5110. It was a young bird when it arrived, but when it began to sing in the breeding season the following year, it had a completely unique song. Oops. So it had come from the genetic analysis, showed that it was a conirostris that had come all the way from this island in the south, Hispaniola in the south, to Daphne. We don't know how it got there, but it was a the genetic conirostris. When it took ages to breed, it being big and with a strong song, it managed to get a beautiful territory on the island, but it couldn't get a mate. For all the Fortis on the island, which was nearest to it, it was the wrong size, it had the wrong song, and so was unable to get a mate until the very end of the second year. And then a female Fortis lost her mate and went into this beautiful territory and mated with this conirostris and produced quite a large number of offspring. We followed all these offspring. They all showed genetic transmission from 5110, and all the males sang 5110's song that they had learnt when they were young birds. Then along came the 2.5-year drought that Peter told you about, and 90% of all birds on the island died, and all these birds went out except for these two, a brother and a sister, an inbred brother and a sister. Well, 2.5 years later, when the rains came back, the brother and sister bred with each other, produced 26 offspring, all but 9 survived. We had a daughter breeding with her father, a son breeding with his mother, and the rest of the brothers and sisters breeding with each other to produce more offspring, which bred with each other to produce more offspring. All birds showed genetic transmission from 5110. All males sang 5110 song, and all were large like 5110. This is just to show that the inbreeding coefficient went up, but still, even now, after 6 or 7 generations, there's still quite a lot of genetic variation. But what was really astonishing was, and this is, the blue dots are what we now call the big bird lineage. They don't lie exactly between Fortis and Conirostris here, but they're displaced. So they have a big bill or big beak on a relatively small body. So you imagine a big head on a relatively small body. And they love tribulus seeds. They eat a lot of tribulus seeds. They go around with Magnerostris eating tribulus seeds. So we were wondering how efficient they were compared with Magnerostris. And we measured the cracking time of these seeds, and we found that they were just as efficient as Magnerostris. And if there was another drought where tribulus was left behind, they would likely do rather well, because not only are they very efficient at cracking tribulus, but that having a smaller body, they would need less food. So this new lineage is behaving like a separate species. It's much larger than Fortis, its nearest relative. The yellow bill you shouldn't pay much attention to, because it just means this was a young bird, and they sometimes have yellow bills when they're young. In morphological space, when we look at bill length over bill depth, Magnerostris lies up here, Fortis lies down here, and the big birds lies in between. And this is the gap that opened up when Magnerostris outcompeted the large Fortis that Peter told you about during the drought. And the big birds moved right in. So it differs in song from all other species on Daphne. Here are 3 generations of the big bird song, very different from Fortis song, Scandin song, and Magnerostris song. They breed in one part of the island. The blue dots are the territories before the drought, the red dots after the drought. They hold contiguous territories, and these territories overlap with Fortis, Scandins, and Magnerostris, who they ignore and who are ignored by them. So in all respects, this new lineage is functioning as a separate species. Will it die out through inbreeding depression? It is very inbred, but there's no sign of that yet. Will the genetic variation be augmented through learning another species' song and gene exchange? It might, but there's no sign yet. So whether it survives or not, this new lineage gives us insight into how a new species can arise and either persist or become extinct. So in summary, when we started on the island in 1973, there were only 2 species, Scandins and Fortis, completely separate. Then came along this unusual El Nino, and the hybrids, the few hybrids between Fortis and Scandins survived and began to breed and backcross, and Scandins and Fortis became closer. Magnerostris came into the island and established a breeding colony then, and then they outcompeted the largest Fortis in a 2.5-year drought, and the big birds moved right in. So we've been able to show 2 methods of speciation. One we think of as Darwin's, exactly as Darwin had envisaged it and described it, and it's now called the allopatric species concept. But also we showed the importance of introgression and how an immigrant Conirostris bred with the Fortis and produced a new lineage, which is completely distinct from all other birds on the island. Now, how wide is this introgression, hybridization, gene flow between species? Well, it used to be thought it occurred in plants, but otherwise it wasn't very important at all. But now, since in the last 20 years, it's now shown to be really important and very widespread. And it's shown to be a source of variation for adaptations to new environments, which can take off on a new trajectory, leading to a new species. We see it in bacteria, in horizontal gene flow, and also in all animals from bacteria up to even humans, where we now know that hybridization and integration in being a source of variation used in adaptation to new environments was vitally important in our own ancestors, with integration between Denisovans, Neanderthals, and early human lineages. It's also very important in cancer, and I can talk about this if anybody's interested. But I think we all know that Darwin's very famous diagram, which he drew in his notebook when he was contemplating how did species interact with each other, and he drew what we now think of as a phylogenetic tree, and he wrote, I think, would he also join up a few of these twigs and put, I now realize that integration is also very important. So I'm going to end with just his very, very beautiful sentence, poetic sentence, his last sentence in the first edition of the Origin of Species, where he writes, whilst this planet has gone cycling on according to the fixed laws of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Now, we are losing these endless forms, most beautiful and wonderful, unfortunately, at an alarming place. We know now, not with our own work, but also with other people's work, that environments and populations are dynamic, they're constantly changing, and for a healthy environment, we must keep them both capable of further natural change. Thank you.
Well, Peter and Rosemary, thank you so very much. This is obviously one of the most famous case studies in all of evolution, and it's a privilege to have heard and seen some of the data that you've collected over the last few decades, which support Darwin's work, prove his genius, and give us a more modern understanding of that discovery that he made. To the audience then, let us virtually applaud. I know that the grants can't hear us or see us, but I'd like you to imagine, Peter and Rosemary, that we are all raising a toast to you and applauding. And yes, I can see some people are raising their hands in the participants list as well. Now, the question and answer session. I have here a list of questions that are just starting to trickle in. And so I'm going to take the ones that, that appear. And Peter and Rosemary, I think you've been gracious enough to say that you'll answer a few of these questions for us. We will aim for a 8:30 cutoff approximately. So this one is from an anonymous attendee. Please do let us know your name so that we can hear from you. What is the most significant risk if you didn't quarantine food or water before going to the islands? Yeah, so Peter and Rosemary, how would you respond to that?
Shall I respond first?
Go ahead.
Well, there's one very big risk. Mostly it's insects, but there's one, yeah, so any insects like fire ants, bringing fire ants to the island, these we're particularly concerned with. There's also another fly that is causing a great deal of damage on some islands that goes into the nests of birds. And so we would have to be, we're very careful not to actually bring any insects like that. Or the other thing is, which is seeds, any seeds of plants or that can take over, because there are on lots of islands, there are uninhabited islands, there are plants that have been introduced through seeds.
Yeah,
so could you?
I would just emphasize the fact that it would be the introduced animals and plants, the fire ants on the one hand, and introduce weedy plants on the other, that would be the biggest threat. many organisms can get around the archipelago very easily, either windblown or flying by themselves. But there are others that are more sedentary, which could be transported. And we're trying to keep that down, invasion of those to an absolute minimum.
Of course. A question here from Paul Johnson. Do you intend to go back to the islands? And if so, when?
We should have been there now. Yes, we intend to go next year. Yeah.
Wonderful. And do you, presumably you have further research ideas? Are you continuing with collecting your data set, or do you have new ideas still?
We always have new ideas.
We always have new ideas, but I think our collaboration with our geneticists, and we're wanting to, to do a little bit more of this to understand the genetics, the underlying genetics, and that's one of the reasons that we're going back.
I think the focal question that we would like to address is what has happened to the big bird lineage since we were last on Daphne. Yeah. And we have plans, I don't know when they're going to be realized, but we have plans to go back to the island and collect samples and find out what their fate is.
Thank you. Next question is from Mark and Janet Trow. I hope I've pronounced that correctly. Are the finch populations across the island impacted at all by migrating birds competing for their food sources? That was a question close to my own heart. I was wondering about the degree to which migration patterns must muddy this picture considerably.
Yes.
To some extent, maybe, but I think the numbers of migrating birds, at least the terrestrial birds, are not sufficient to really make an impact. Now, if you mean I should qualify that. I'm talking about migrants from outside the archipelago coming through the archipelago, like there are warblers that come through. There's a bird called a bobolink that comes down from, remarkably, from the Canadian Arctic and goes all the way down to Argentina for its wintering. So the numbers though there are very small. But within the archipelago, yes, for example, after an El Nino year of terrific production, there can be a mass movement of young finches from one island to another, and they certainly could impact the food supply of that other island to the detriment of the residents.
Super, thank you. A quick comment here rather than a question from Lottie, who says that she teaches university students, and this was a fantastic overview of a phenomenal long-term study, and thank you very much. I'm sure she'll be using it in her lectures. A question here from Kim Lindsay. Of all of the things you've learned on the island, what has been the most surprising, maybe other than the new lineage, which was such a stupendous discovery?
Well, the new lineage, starting with that one, really was spectacular. And that's something that at the outset we could never have predicted. The most dramatic results that we have found, I mentioned at the beginning, 2 selection episodes. And to step back a little bit, back to 1973 when we started off, we knew that we were working in a dynamic environment, that is climatologically, or climatically I should say. It was an environment where there could be very strong stresses on populations sometimes, but not others. But we did not know that we would be able to document and interpret very strong natural selection so quickly, so early in the study. People ask us, did you plan 40 years of research right at the beginning because you knew it was a wonderful place to go? And the answer is no. We first of all had one field season where we did a survey of populations across the archipelago. And then we concentrated on Daphne because we found that almost all the birds had survived that first year, whereas on other islands, they had moved out of study areas. And little bit by little bit, from one dry season to the next dry season, we increased the length of the study. So by 1977, we found that this selection event and decided from that point on, this really deserves a really long-term study.
And you probably had no idea quite how long that would become, but
oh, no idea. And I have to say that money is a big determinant of how long one can keep going. It does help to have really spectacular results right at the beginning of the study. But that in turn, I think, was helped by the fact that we could see what an unusual environment Daphne was right at the beginning and thought that it had exceptional potential.
Thank you. Another question from Hugh Williams in Shrewsbury. It's been really fantastic to hear about Darwin's original ideas being developed, but he was interested in Rosemary's comment right at the end about cancer and the involvement. I think I got the sense that the genes which had that involvement in controlling beak morphology and that have similar sort of roles in mammals, for example, might have some implication in cancer. Is that right?
Yeah, yes. Well, not quite the genes, but the idea of introgression has a big implication in cancer. In fact, we have been asked to go, or we did go to both the Cancer Research Center in Texas, invited us, and also in London. And I think I can actually go back to that slide. Let's see, yes, this one. Where the idea of cancer clone evolution taking place within tissue ecosystems. And the idea is, and this was very exciting, this was in London, where you can see the cancer in, let's say, a colon and one in the lung, let's say, is taking place in very different ecosystems, almost as though it's on two different islands, if you think of the finches. But the other thing is that there is actually introgression, gene exchange between these cancer clones. And these can often happen, they put out little blobs of DNA, and then this DNA can actually move from one cancer clone to another one, and have a big effect on the cancer itself. So I just put down one, there are many examples, but this is a nice one where there is, well, nice from the cancer's point of view, not from the human point of view, but there's a tumor suppressor gene. And this was destroyed due to rearrangements associated with introgression. This was actually moved across by a retrotransposon, but it's exactly the same idea through horizontal gene transfer, a gene exchange which suppresses a function which leads to tumor formation. And there are many cases now like this where there's an exchange of DNA is happening.
It's fascinating. Another question here, more on the climatological front from Mark Fermor. Have you done any work with modelers on the impact of climate change or ingression of other finches to see how things may progress in the future? So really, this is looking at the future of obviously our climate and how that might affect things.
Well, we've been asked about whether there are signs of climate change in Galapagos, and the answer for the terrestrial environment is no, because the climate swings so much from drought conditions to extremely wet, from cooler to very hot conditions. So given all those swings, it's very difficult to pick out a very slow increase in average temperature, if there is one. In the marine environment, on the other hand, the signal is there. And so we know that the waters are increasing and that the corals are suffering, and maybe some other fish and some fish species as well.
And the other thing that's happening is that there seems to be an increase in extreme conditions. So an increase in extreme droughts and an increase in extreme El Nino years. So it fluctuates between the extremes of getting greater. And the modeling there has shown in the past, looking at it in the past, that this seems to be happening.
There'll be members of this audience who will live for the next 50 years and be able to find out, and more, and find out if the Galapagos are increasing in temperature or drought conditions or whatever. Unfortunately, we won't live that long. But I think it's an open question for people who have ambitions to go to the Galapagos as to exactly what effect the change in the global environment is having upon this absolutely wonderful system, which is one of the few in the world that's kept in close to, not exactly, but close to pristine state, at least on several of the islands.
Well, looking at our participants list, I can see some of the students from our school are here, and I think I would not be at all surprised if some of them grasp that idea firmly and maybe look to do that sort of thing. Another question from another anonymous attendee. This might be the last one we have time for. How similar are the evolutionary processes on other islands closer by and further afield? Are similar effects emerging? Now, of course, the Galapagos have all sorts of unusual qualities and conditions that might make them a fairly unique situation for evolutionary studies, but have you looked at other island systems to see similar patterns?
We haven't looked at other islands, but I can tell you that how difficult it would be if you went to, for example, Hawaii, where 50 species were once existing. We know that from fossil and subfossil material. There are now about 21 species. I don't know what the total is. It's between 20 and 30 anyway, as a result of massive extinctions. So with such an incomplete fauna, it's very difficult to make sense of whatever anybody could study in the short term. That's, in contrast, the big advantage of the Galapagos. So anyway, back to your question about have we studied any populations on islands with a similar intent? No, we haven't, but we do know that other people have studied island populations of birds and found small, very small changes from 1 year to the next in morphological features, and not just in islands as well, on continental regions as well, but they're very, very small and subtle and slow-moving effects. Galapagos finches, on the other hand, in the appropriate environment, can show very strong effects in a short time. I often think of the early studies of Drosophila genetics, and people have wondered, well, why so much effort has been spent on Drosophila? And the answer is that they have very large chromosomes in the salivary glands, which made the characterization of the genetic constitution of chromosomes and the dynamics of chromosomes very easy to study. And so people spent a lot of time studying Drosophila and forgetting a lot of other flies. Now, the Galapagos are similarly very suitable for studying a different kind of problem. And that's why it's yielded so much.
There are other people who have studied. I mean, there's some very nice, well, fish, for example, Icelandic fish in the different fjords in Iceland that are different age groups, and there's been a terrific amount of very similar selection there. And then also Anolis lizards on the Caribbean and places like that.
We're not going to stop, you know. You realize that there's sticklebacks as well in North America that have been studied
sticklebacks, yes,
in great detail, yeah. And very strong selection discovered by a group of observers in Alaska. Anyway, maybe we shouldn't go on forever.
I'm sure we'd all love to hear you go on forever, but it may be that people have made plans for 8:30 and onwards. And I was told that that's when we ought to bring this to a conclusion. So I will just once again, on behalf of everybody here, silently at their computers, offer a vote of thanks to you across the ocean, to your, are you in Princeton at the moment?
We're in Princeton, yep. Yes.
Yeah, and snowy there as it is in the UK, I think. Yes. And to wish you well and good health and to stay safe. And that let us hope in the not too distant future you're able to return to this country and perhaps give us a repeat performance in Shrewsbury. Until then, thank you from everybody here. Apologies if I didn't get all of your questions answered. I think one or two of them didn't quite make it, but I think our time is up.
Well, if anybody really wants to ask questions, we're on email, so they can ask. They can email us. Yeah.
Well, thank you very much.
And thank you very much, yes.
And on behalf of the Darwin Shrewsbury Festival, thank you for coming. And just a reminder to look at the website. There's an awful lot going on this week, over the next few days, particularly talks and virtual exhibitions. Please do book yourself into those. And I hope to see you there virtually. Thank you very much for coming.
Thank you very much.
Bye-bye.
Bye-bye.
Bye-bye.